All right. So hopefully that was ample time to knock out the warm-up. Let's go ahead and start with the lesson for today. We're going to start with some quick practice. We did talk about this at the end of yesterday's lesson. Yesterday, we talked all about transcription, so the process of taking DNA and transcribing it to RNA. Now, the thing that we have to remember about transcription is that when we're transcribing from an A, we don't go to a T, because RNA does not have T's, it has U's. So we transcribe an A to a U. All right, so keep that in mind. So this first letter here, I'll knock it out for you all. When I transcribe this A, it becomes a U. What's next? What is T transcribed to?
A.
A, good.
T, A. Oh, U. I forgot.
U, A, yeah. No
Right.
T's in RNA. Okay, keep going.
C,
C,
G, A. C, G, U, A, G, G, U, C.
Excellent. All right. So this is, again, the process of transcription, going from DNA to RNA. So good job with that. We did have an activity yesterday that asked students to practice with this. So if you're a little confused, I would encourage you to go back and take a look at that assignment if you haven't done it yet. And because the process of transcription and the process of translation can be a bit confusing, so I want to take some time to remind you all, I do have office hours available to you. So if at any point you'd like to talk, we do have that option, and I can walk you through some of these more difficult concepts. Please do keep that in mind. Okay. All right, so today we're focused on translation, which is the second step in the process of protein synthesis. At this point, you should be taking your notebook out. You should be opening it up, getting ready to write. You should record the title of the lesson, which is Protein Synthesis, Translation. Today's date is October 9th, 2020. It's Friday. I know we can all be grateful about that. And it is the fourth day of our fifth unit. So Unit 5, Day 4. Our objective for the day, biologists will be able to explain the steps involved in the process of translation, in which mRNA codes for a chain of amino acids. The essential question: how does a sequence of nucleotides code for a sequence of amino acids? So what you'll see is that I've italicized and bolded the word sequence for a very important reason. And that's, again, structure determines function. In this case, sequence is structure. So if I change the sequence, I'm going to change the function. We'll talk more about that later in the unit. So please make sure that in order to take the best, most efficient, most effective, and most organized notes, you've written down the lesson title, you've written down today's date, and you've written down the day of our unit, Unit 5, Day 4. I'm going to give you 5 more seconds to finish writing. Okay, great. So the central dogma of biology is a phrase we introduced yesterday, but it is so important because it describes the process by which we go from DNA all the way to proteins, and proteins determine traits. Proteins determine an organism's traits. So yesterday we talked about the first part of this process, going from DNA to mRNA. Today we're going to talk about the second part, going from mRNA to amino acids, and this is a process called translation. Okay. The central dogma of biology is so important because it describes the process by which genes code for proteins. And of course, we pass on our genes from one generation to the next. Genes are the instructions. They are the instruction manual that tells our cells what to make, when to make it, how to make it, and what quantity to make it. And so our genes are so important, but this process allows us to go from genes, which are DNA, all the way to proteins. So keep in mind, the words that are highlighted are the words that you should be writing down. The second step, that is the focus of our lesson today, is translation. The goal of translation is to read the mRNA sequence, the messenger RNA sequence, and begin linking amino acids together to make a protein. So we're going from mRNA to amino acids. This process happens inside of ribosomes, which makes sense. From Unit 2, when we first introduced ribosomes, we've been talking about the fact that they are the protein factories. They make proteins for us. So here is an in-depth discussion about how that happens. Now, it's important to remember that transcription happens before translation. Transcription is going from DNA to mRNA, and then translation is taking that mRNA and going to amino acids. Transcription happens first. We can remember this because the words are in alphabetical order. Transcription is alphabetically before translation because C comes before L. All right. Are there any questions about this slide so far? Okay, no questions. So what I want to do now is give you all 3 minutes to write this slide, focusing, of course, on the words that are highlighted. I'm not going to talk for these 3 minutes. I just want you to write. Then at the end of those 3 minutes, I'll explain what is going on this slide. Okay, so just take 3 minutes now to write, starting now. Okay, just 30 seconds left. Try to wrap up writing. Make sure you're focusing on the highlighted words. Okay. So let me explain these steps now that you all have written them down. So if we think about the end of transcription, of course, transcription comes before translation. So the end of transcription ties directly into the beginning of translation. At the end of transcription, we have an mRNA strand that leaves the nucleus and it goes into the cell's cytoplasm. Then the first step of translation is for a ribosome to come and attach to that strand. Okay, so the two steps, the last step of transcription and the first step of translation, flow back to back. We've got to, even though we talk about these as two different processes, this is all happening stepwise. So a ribosome will attach to that mRNA strand. Then the ribosome will read that mRNA strand in groups of 3 bases, so 3 letters at a time. We call these groups of 3 codons because they code for something. They code for either an amino acid or some other message to the cell. That other message might be a stop message, which we'll talk more about, but it just signals the end of translation. I shouldn't say transcription. Sorry about that. So the ribosome reads that mRNA in groups of 3 bases called codons. And then as those codons are read, there's a tRNA, a transfer molecule, that will bring an amino acid to the ribosome based on the codon that's there. So each codon is going to code for a specific amino acid, and that amino acid gets brought to the ribosome by a tRNA. And then the last step is that the ribosome will start to link those amino acids together to start making a polypeptide chain. Poly is a prefix that means many, and so a polypeptide chain is a chain of many peptide bonds. Eventually, this will become a protein. Okay, so we're going to bring amino acids to the ribosome, and the ribosome is in charge of linking them together. It starts with an mRNA, and it ends with a chain of amino acids. This is translation. Okay. So this image is an image of translation. We've got all those terms that we saw on the last slide here. It starts with your mRNA strand, which is this red strand here. Your mRNA strand is made up of all these nitrogen bases, C, G, A, C, U, G, C, A, U, G, C, U, G, A, C, A, G, U, G, C. But we don't read them one by one. We read them in groups of 3s called codons. And when I say we read them, I'm talking about this big blue ribosome. Okay, so the mRNA strand pulled through this ribosome, and the ribosome is reading these codons. Each codon fits perfectly well with a tRNA because each tRNA has a specific anticodon. You don't need to worry too much about that, but you can see in these anticodons that the nitrogen bases are paired. U is paired with A, C is paired with G, A is paired with U, G is paired with C is paired with G, with U is paired with A, so on and so forth. So each codon has a partner anticodon that they fit perfectly together. And so this tRNA molecule is bringing with it an amino acid. It's the ribosome's job to link the amino acids together with peptide bonds, and eventually those peptide bonds become a polypeptide. Okay? So those are the steps of translation. Are there any questions at this point? I understand this can be a confusing process. I don't want you all to be too shy or reserved to ask a question. Questions are good. The smartest people ask the most questions. We also have the best attendance we've had potentially ever. I'm going to give people 10 seconds to think about any questions they may have. If you don't want to ask them right now, write them in your notes and you can send me an email or a remind message, or you can schedule a time to talk with me one-on-one and we can make sure to get those questions answered. I don't want you all to be confused about anything. So 5 more seconds, think about some questions. All right. So the steps, once again, the ribosome attaches to the mRNA. Second step is for the ribosome to read the codons. Those are groups of 3. The third step, the tRNA is going to bring its amino acid into the ribosome. And then our last step, the ribosome will start to assemble a chain of polypeptides. And that will eventually become a protein. So translation is, when we talk about it in terms of protein synthesis, when we talk about it in terms of making proteins, it's very similar to when we talk about it in terms of language. To go from one language to another, to translate from English to Thai, to Vietnamese, to Scandinavian, to Spanish, to French, to Mandarin. Okay, you can translate these languages just like we can translate between the language of nitrogen bases to the language of amino acids. And our first step, what we talked yesterday about, was transcription. We were in the same language. DNA and RNA are both written in the language of nitrogen bases. A's, T's, C's, G's, and U's. They're just slightly different formats because DNA doesn't have U's, it has T's, and RNA doesn't have T's, it has U's. And now we're moving away from the language of nitrogen bases and we're going to the language of amino acids. So that's a different language. That means it's translation. Just like we can translate this from Hebrew to English, which says that you are the most amazing students ever. You can thank Miss Baxley for that. Not gonna watch this right now. We watched it yesterday. So this is a codon chart. What you're seeing on the right is a codon chart. And the codon chart has all 20 of the amino acids in it. Please make sure you write down the words that are highlighted. So in nature, we have 20 amino acids. And each of those codons that we've talked about, the groups of 3 letters, codes for a specific amino acid. Every codon codes for a specific amino acid. We use these codon charts to help us determine what amino acid we're going to get from what codon. So if I give you a codon, you should be able to use this chart to tell me which amino acid that codon codes for. Okay. So the codons will either tell you which amino acid you're going to get, or they code for a stop message, which tells the cell, which tells the ribosome to stop translating. Okay, and that stop message is either UAA or UAG. That's your code. Those are the codons that code for that stop. We'll talk more about that. But there's an order to determining which amino acid you will get. So you first need to look at the first base, then you need to look at the second base, and then you look at the third base. Your first base is on the left side, your second base is on the top, and your third base is on the right. We're going to talk more about how to read this chart specifically. But this is why the sequence is so important, because if you change just one of those letters in your codon, you could potentially end up with a different amino acid. So our cells have to be careful, and this is why mutations can be so dangerous, because even changing a single nucleotide, just changing one of these bases, one of these letters, can totally throw off the protein that you end up with in the end. All right, so keep that in mind. Sequence and structure matter for the function. If I change the sequence, then I'm going to change the function, potentially. All right, we have two different codon charts. Either one is okay to use. It's just a matter of preference. When I was in high school, I was taught to use this chart on the left, and then in college, I learned about this chart on the right, and it became my preferred chart. So I choose to use that one most of the time now, but either one is applicable. They give you the same results if you do things correctly. So it's a matter of preference. Let's talk about the chart on the left first. So it may be a good idea to write these directions down. I will give you 2 minutes at the end when I'm done talking about it to write these directions down. So the first time you might just want to listen to me talk so you can make sure you know what's going on. So in order to use a codon chart, first you need to know what your codon is. And remember, a codon is a group of 3 nitrogen bases. In this case, I'm telling you that your codon is GAC. GAC. So the next thing that I need to do is identify what my first letter is. Can someone tell me what the first letter in this codon is? It'd be real simple.
G.
G, thank you so much. So that means that I need to look on the left side, and G is red here. Let me use some colors to help you all see this. So I start on the left side, always. That's where I start with my first base. And I see that there's a G here. So what that means is that I should be focused on that row. I should be looking in this row right here, focused only on the G. All of this stuff up here no longer matters. Okay, I can ignore this stuff. I'm only focused on what's in this row now because G is my first letter. My second letter, of course, is A. So now I should be looking in at the top of my chart, second base. My second letter is not U, it's not C, it's A. So I need to be looking in this column here. But keep in mind, I'm also looking in this row. So I'm looking at the overlap between the two down here. Because I don't care about any of this stuff up here because my first letter was G. And I don't care about any of this stuff over here or over here because my second letter was A. So I'm just looking in this small section here. What's my third letter?
That is C.
C is the third letter. It's the third base in the codon. Good job. So once I've realized what my third letter is, I have to look on the right side now with my third base. And I see here's C right here. So I can draw an arrow from that C over, and that tells me that my amino acid is ASP, which stands for aspartate or aspartic acid. So this is how you use this codon chart. You first need to know what your first base is, and you find the row for that first base. Then you need to know what your second base is, and you find the column for the second base.
This is confusing.
Okay, we're going to get more practice with it. I appreciate the honesty.
No problem.
Who is that?
Anthony.
Okay. Anthony, I want you to work with me on this next one.
Okay.
Maybe you'll enjoy this next chart better.
Okay.
So what's my codon in this example?
GAC.
Excellent. So this is the same codon that we had in the last example, which means that if we do everything correctly, we should end up with this exact same amino acid. Okay, so keep this amino acid, ASP, aspartic acid, in mind.
Okay.
What's the first letter of my codon?
G.
G. So in this chart, I want to start in the middle.
Okay.
Should I look in the top right, the top left, the bottom left, or the bottom right if I know that my first letter is G?
Top left.
Top left. Okay, so I'm starting in the middle and I'm moving outwards. Now, Anthony has told us that our first letter is G, which means that I should only be looking in that section there. Nothing else matters. I can literally ignore it. Okay, so now I'm literally only looking at 1/4 of the pie, and I can ignore everything else.
Okay.
Anthony, what's my second letter in the codon?
A.
Okay. So should I be looking here then?
No. The
Should I
third
be
one.
looking here?
No.
Should be looking right here, exactly. So that means I can ignore the other sections because I'm only worried about my second letter being A. Remember, I started in the middle and I'm moving outwards. So G, A,
Then C.
and then C is my last letter. Good.
It's gonna be aspartic acid, right?
It's going to be aspartic acid again, which is what we got last time.
Yeah.
Does that one make a little bit more sense, Anthony?
Yes.
Okay, good. So we end up with the same amino acid, which is exactly what should have happened because we have the same codon. So I prefer to use this chart. It's a little bit easier to, you're not as likely to make a mistake with this one, I think. You start in the middle and you move outwards.
What does GAC and stuff like that stand for?
It doesn't necessarily stand for anything. It codes for this amino acid. So if I go all the way back, you can see in this chart, I have all these different amino acids around here. I've got 20 different amino acids.
Okay.
But GAC only codes for one of those amino acids.
What's the U?
GAU codes for that exact same amino acid, which is, I'm glad you noticed that. So what Anthony just pointed out is the fact that you can end up with the same amino acid even if you have a different codon. So GAC gives you aspartic acid, but GAU also gives you aspartic acid.
Okay.
All right. So let's do some more examples. I'll do this first one. So my mRNA codon is AGU. So that means my first letter is A. So I'm only looking in this row right here. I'm just looking in that row. Everything else I can ignore. If it's not in that row with my first letter A, it doesn't matter right now. My second letter is G, which means I need to be looking in this column. So I'm looking in the overlap right here. Nothing else matters, just this section. And my last letter, my third letter is U. So here's U right here. So I need to look right here, and that tells me my amino acid is serine. Okay, somebody else, someone likes to volunteer for this next one? I'll help you if you get stuck.
The mRNA one. I mean the mRNA one. I can do it.
Joseph, you're gonna help me?
Yeah.
Okay. So now we're looking at GAC.
All right.
So which row am I looking in? The first row, second row, third, or fourth?
The last row.
The last one down here. Which column? The first column, the second, the third, or the fourth?
Third column.
The third column, because my second letter is A, which means I only have two options, aspartate or glutamate. Which one is it?
Oh, it's aspartate.
It's aspartate. And how did you know that?
Because it has a C at the end of the third letter.
Good, because my third letter is C. So I'm only looking in this row right here. Good job. So aspartate is the correct answer. I know you guys can't. Okay, there it is. Aspartate. Thank you, Joseph. Now, if you're like, I don't get this chart, but the other one works for me, that's perfect. It's a matter of preference. If you prefer this one, then you can go ahead and work with this one going forward. All right, would anybody like to try CAG? What does CAG get us? Does anyone know what CAG gets us? Oh, excellent, Jessica. Good. We end up getting glutamine. So first letter is C. So I'm looking in this second row. Second letter is A. So I'm looking in the third column again. So this section right here. So it could either be histidine or glutamine. But my third letter is G. So I've got to look down here. It's glutamine. Good job. All right, next one. AUC. What is AUC? Yeah, anyone can answer this one. AUC. Good job, Jessica. Good. So we're working with isoleucine in this case. A is my first letter, so I'm looking in this general section. Everything else I can ignore if it doesn't fall in this general section. My second letter is U, so now I know I'm looking in an even smaller section. So I could have, excuse me, either isoleucine or methionine. But C is my third letter, which means I've got to look right here. It's isoleucine. Good job. Okay, now for the people who prefer this chart, can we work with AGU? If the codon is AGU, then what's my amino acid? Oh, somebody sent a message. Thank you, Jessica. You're killing it right now. That is correct, serine. So we want to start in the middle and move out. So my first letter is A, so I'm looking here now. My second letter is G, so I'm looking over here now. And my third letter is U. So I'm looking right here, and that tells me it's serine. Lastly, GAC.
Aspartic acid.
Good, Joseph. Yeah, so that's the same one that we started with. Okay, so this is GAC. That's got to be aspartic acid, also known as aspartate. All right. So we want to put it all together now. Yesterday, we talked about transcription. We transcribe from DNA to mRNA. And today, we've introduced translation, where we translate from mRNA to amino acids. So we want to do both of those things here. So if I am transcribing T, what does it become? What does a T become? What is T matched up with? Not U, but I'm glad you realized that RNA does not hit M, Jessica, so that's good. But it's not U, it's
What's
A.
U?
Good, thank you, Jada. Now A, what does A get transcribed to? Now it's your time to shine, Jessica. U, good, good, good. Thank you, Jaden. Lots of U's. All right. What about C? What does C become? It'd be a little bit easier if you guys could just unmute yourself. Thank you. All right, G. What's next?
C.
Now we've got C. Yep, C.
C.
What about G?
You got another C right there.
Another C.
And you got G.
G.
A.
A, good.
T.
Ah, no T's in RNA.
Oh, I mean U.
Yeah, U, good.
And then you got C.
C.
And then you got another T.
You did it again. No T's in RNA.
U. I keep tripping with the A's and
U. Okay,
and
good.
Ooh.
That is tricky. That's tricky. So you would be right if we were only talking about DNA. You'd be
Hey,
right, but we're transcribing to RNA. And now we finish with two A's. So good job. Thank you, Jaden, for that help. Or was that Joseph?
That was me.
Joseph. Thank you, Joseph.
That was me. Don't it?
Okay. Jaden, Joseph, and Jessica, I appreciate all three of you. All right, now we want to do the process. We want to do the work of translating. So we are currently in the language of nitrogen bases. I've got AUG, CCG, AUC, UAA, and conveniently, I've already put them in your codons. So they're already in groups of 3. So it makes it pretty easy to see. So AUG, what amino acid is that? You can just give me that abbreviation. What amino acid is AUG?
It's a T.
It does not start with a T, you know.
U.
We're looking for, yes, thank you, Jessica. So we're looking for the amino acid. So this is where you have to use your codon chart. Yeah, sorry, maybe I should have explained that. So at this point, we have our codon right here, AUG. So my first letter of the codon is A. So I'm looking in this general section.
Met.
My second letter is U. So I'm looking in this smaller section right here. And then my third letter, my third base is G. So that means I'm looking down here at the bottom. That's MET, that's methionine. Good. What's next? My codon is CCG. What does that give me? Good. Thank you, Alyssa. Thank you, Jessica. We've got proline, PRO, pro. Now I've got AUC. Instead of AUG, this time I've got AUC. What does that give me? Okay, Jessica, she's got this down now. ILE, that's isoleucine. And then UAA, what does that get us? Be careful. Yes. All right, so this last one, UAU, Jessica just told us it is stop. So let's make sure that's right. So the first base is U, so I'm looking in this general section. My second base is A, so I'm looking in this smaller section. So it can be either tyrosine, or a stop codon. And my third letter is A, so it is indeed the stop codon. And that stop codon is telling the ribosome, okay, that's it. This is the end of the chain here. Break it off. So it's an important codon there. So excellent job. If you're still confused, we have a lot of practice available, starting with the exit ticket. And there's also an activity, a translation practice activity. I want to show you something before I let you guys start working on your own, though. So I'm going to share my Canvas tab. So I'm in our Canvas page. I'm in our 5th module. If you scroll down to Unit 5, Day 4, you'll see that there's a codon chart there available for you. If I were you, I would save it to my Google folder as well as bookmark it. All right, so if you click on it, there's a link there. That link will take you over here. All right, so you've got both charts available to you. Whichever chart you prefer, you're more than welcome to use. I'm going to go back to Canvas. Go back to our module, 5th module. Okay. So the assignment for the day is called Translation Practice Edited. You've got both codon charts available to you yet again here at the top of the page. All right. What you're going to need to do is transcribe and translate. Some of the questions will ask you to go from DNA to mRNA and then to amino acid. Some will ask you to go backwards and so on and so forth, but you guys will see what I'm talking about. The last part of the activity is a gizmo. So we talked about gizmos a couple of days ago. But if you haven't created your profile yet on Explore Learning, then you will need to do that before you can finish the gizmo. The gizmo is called RNA and protein synthesis. You just need to complete the activity and answer the assessment questions. Okay, so you might not get all of that done before class ends, but you should at least be able to get done the exit ticket and the first 2 questions of this activity. We've got 21 minutes. I'll be here on mute if you have any questions about gizmos or about this process. Jaden, you are not dumb. You've been really helping us out here, so I Makaran, I see your question. Give me one second. The class code is FNNJ5K. I'll send that to the chat. FNNJ5K.
My computer had died.
Okay, no problem. Let me know if you need any help with the assignment.
Okay.
Okay, folks, it is 1:20 at this point, so I want you all, oh, I'm sorry. Will I upload the live? Yes, I will be uploading the live recordings. Most of them have been uploaded. I'm in the process of creating a YouTube channel to put the rest of them up. Thank you for that question, Jessica. But it's 1:20 now, so I want you all to go ahead and get ready for fourth block. You have one more class, and then you're headed into your weekend. I hope that you have a productive, fun, and safe weekend, and let me know if you have any questions between now and Monday. I will talk to you all soon. Adios.
Have a good